First, I want to thank the organizer, and because they did a great job. I think it has been a fantastic meeting, and well, also for the invitation. So this is the original title, and I think I will deviate a little bit about that. But basically what we're interested in is not too much about biology, evolution from Luca, but what happened before, and especially what happened at a time where you have very likely no nucleic acid-based information system. And the problem is in this case that we have schizophrenic, so to speak, behavior in the lab, because on the one hand we want to show certain processes, so we will create chemical system to show those processes, and on the other hand we are interested in understanding, given our knowledge of the chemistry on the early Earth, whether or not certain general biophysics and chemistry apply to this molecule and set of molecules that could have been on the early Earth. So basically you have seen this figure at least 3 times, if I'm not mistaken, or 2 or 3 times. I use it just to locate where we are. So we have prebiotic chemistry, that was Matthew Poner yesterday and other people, and then we have at some point life appearing. The question is what happened in between. Well, logically, what happened in between over time should lead to something that looked like a cell, otherwise we wouldn't be cellular based. But the question is what happened before. And people that have been doing research here have tried to come up with names and ideas how those systems that preceded cells should look like. And the first name was actually Protocell, was proposed by people like Louisi, Dave Dimmer, and others. And actually it's quite a complex system, so I prefer to speak about minimal self-replicating chemical system, because here it's simpler and it doesn't have the taste of cellular behavior too much. But the basic idea behind that is we want to create in the laboratory system that could mimic at least some part of the systemic and functional properties of living system, and could be model, I say model, and I repeat for the third time, model, please, of system that might have existed on Earth and preceded cellular life as we know it, and basically Luca. Now the big problem we have in this field is to agree about anything. And one of the biggest questions, and I'm sorry I missed the talk about the definition of life, is what is life? What are the minimal requirements for life? And obviously everybody choose an operating definition in my case, and don't take it too literally, because I use words from biology to make the people understand what I mean by that. I think we need a triad of molecule or function, an information molecule that either define or basically control a metabolism. Here, as a chemist, I would prefer to speak about reaction network, but most of the biologists will say, what is that? And we have some form of container. And what we hope is if we create a system as complex as this one, we might have emergent property that life knows for a long time, self-maintenance, self-replication, and under constraint a form of evolution. The problem, those systems are very simple. So even if we are dependent on the environment today, those systems were extremely dependent on the environment. So in fact, environment is a 4th part of the system. So the next question that interests us is basically, when does it occur? How does chemistry get into biochemistry? And here we propose at some point a kind of scenario that in our point with Peter Valde tried to summarize what we know today. And you can imagine, as Bob Eisen, or Robert Eisen, I'm sorry, was presenting, at the beginning you have some organic on the surface, mineral surface, you start some simple chemistry. At some point, these systems have to become dispersed. That's a big step, actually, to maintain stability and integrity of the system in the dispersed phase. This is a big trouble, a big transition, but still, these are very simple things that don't look like cell. And then as you evolve more and more, you must get to a structure that has the blueprint of a cell, obviously, because at that point, we are still not alive, but we are preceding living system that everyone in this room likely will recognize as cellular living system, even if they are not exactly as exquisite as a contemporary cell. So the problem is here, this part is very long, so it's actually way longer in my view than this stage. When do we get systems that start inheriting traits from previous system? Because it's very important to inherit traits from a previous system. It allows you to build on and to increase the complexity. But when I say inheritance, please don't look at the Darwinian tree of life where you have branches that are almost linear going up from the bottom. Here, I think the inheritance is more stochastic. But basically, at some point, it becomes important to have series of systems that have the same basic function properties and start being able to increase their complexity this way. So information inheritance, and there the biologists will kill me, but still I'm a chemist, so I don't care too much. In generally, the chemists say, well, there are 4 forms of information, and you see the various parts. So it goes from the atomic composition, bond composition, configurational information, S and R, and functional information, well, whether a molecule has the right redox potential, for example. And then down there, we have very precisely defined biological information, which is an information encoded in a sequence-specific polymer. Now, the problem is a bit the information in chemical system. Actually, chemical systems are, how long? 10 years old, officially? Otto? I think about 10 years old, the word chemical system appears as a field. So the question here is composition could be something, architecture, shape, or function. And we are interested in this case in saying, well, those systems are built to somehow be able to self-replicate and transmit their property and get enriched over the generation. And what is necessary and sufficient to define information replication in chemical system? And I'm not saying I have an answer to that question today. How does self-replication, which is an important process that we are interested in a chemical system, relate to the emergence of trait inheritance? That's another problem. We're just starting to scratch the surface. Now, the first big system, so you all know the RNA world. I think all of you have heard about it. But this is not only the RNA world, there is the metabolic world, and there is the lipid world with the concept of composome. And basically that's, in my view, the first attempt to try to figure out how information of a system can be inherited by daughter or son system. And this is based on self-assembly. Because obviously, if you have no information molecule that is specific, like a nucleic acid, well, you have to have a way to get a composition, so to speak, frozen in time in some way. And this is the idea of the composome. And obviously, in the origin of life, if you speak about self-assembly, you have to ask how many molecules could have done self-assembly. So is it realistic to speak about population of aggregate that would be so different that you can say that's one unit, that's another system, that's a third system? The good thing is actually we have a lot of fulfil at our disposal. So, oops, went wrong. Some are found in chondrite, some are done by so-called prebiotic synthesis. And from the chemical point of view, if you look at the various functions that are there, this is quite a large number of functions that chemistry knows. And all of them, to some extent, have what we call amphiphilic qualities, so basically could self-assemble. So, two things to not forget is the composome is actually looking at something very real. Of course, today we have lipid, and lipids are basically formed principally from fatty acid. That's 90%, I think, of the membrane. If you catabolize all the possible head group together. And this is obviously there, but in a world defined only by chemistry and physics, whatever component can self-assemble will self-assemble, and that means the richness of a membrane that would be prebiotic is much larger. That means that there is very likely a detectomy, detectomy between the research that is done with one lipid or one amphiphil and what life was really about, or pre-life or system chemistry was at the time. So does it work? This idea that the composition of a system can be somewhere frozen and pass to further generation? Well, some systems do that. Here we have two new, I just wanted to make a remark for those who have no knowledge of amphiphil. Amphiphil are basically molecules that self-assemble, but they don't self-assemble directly. What we have is, first, when you start suspending amphiphil in water, what you do is you suspend what we call monomer. By reference to polymer and monomers, actually these are single molecule. And at some point you reach a threshold value for vesicle is a CVC, a critical vesicle concentration, at which the monomer stop being added. So you don't have more monomer in your system. If you add more molecule, they will start forming structure. But very important, those structure and your cellular membrane today too are always in equilibria with the monomeric species. So monomer go from this state to that state at every second. The only difference with our membrane, lipid membrane, this value is around 10 to the minus 9 to 10 to the minus 12 molar, so we don't notice it. But in this case, very simple amphiphil, it can go way higher in the 10th of millimoles and even larger. So that was my first question, is if we have a set of amphiphil, can we get basically a population where we have a diversity, and this diversity can be inherited? Well, we can see with new amphiphil that we synthesized with John Sutherland and Terence Key in England, both in England, that, for example, the phosphate, acyl phosphate and the acyl phosphonate in this case are amphiphil that are amphiphil, but alone they will not form any structure. They need actually what we call co-surfactant. In this case, both of them can, for example, use the starting material, which is an alcohol. The interesting thing is then, if you add more and more material and your structure grow and start dividing, well, if the new generation exists, it's bound to have a very similar composition. Because if you were to lose one of the surfactants, the system falls apart. So here it seems composome working. Well, let's take another example. This is the total series that was found up to the 90s in fatty acid in meteorite. So basically, this should be for surfactant. What happened if we mix those systems? Well, here we have done an experiment where we mix 4, 5, 6, and 7 systems. Actually, even where we mix all of them, but I will only present those 3 sets. And we asked the question chemically, what is the composition of the monomeric unit in our system when it's at equilibrium. And what we discovered to our amazement is actually, as you increase the number of species, and you have to know that the amphiphilic character decrease in this direction, well, what you discover is actually some molecules are necessary, but they are not participating to the formation of membrane. So basically, they improve the formation of structure by the larger member of, or the longer member of the community, but themselves, they are present as monomers. So in this case, the system identity is dependent on the environment, so you will have a problem transmitting identity traits here. The second and last thing I wanted to say is, well, let's assume for 1 second that we have our system that are basically resilient, can pass on their own composition. What happens over time? Do they stay in one form, or do they start mixing together? Because this equilibrium seems to indicate that this is the case. And actually, every experiment we have done, looking at, for example, how systems behave under temperature, making two populations and seeing how the two populations fuse, all point out that within a few days, the mixing will be so intense that the composome has no way to to exist in his idea and to answer our traits. Our research basically, it's do we have traits inheritance. So what we did is, well, look at the next level of complexity, and that's the interesting thing with a chemical system, is we can do function, and that becomes very interesting. Here you have the shape system, but since I have 5 minutes, I won't go too much. This is a logical way of looking at the chemical system. We are looking more at the way that a membrane, for example, itself could be a chemical system. The reason being that it's very difficult to encapsulate material in a vesicle that looks like a cell. It's much easier to get material to interact with a membrane given the right properties. So now the question is, can we find or design systems that are functional and basically pass their function to the next generation? Because that would be a way to establish a form of inheritance. So I will pass this one. We have here the system that's a rather non-prebiotic system, but the idea is to use light energy to basically power the formation of an amphiphil from a non-amphiphil system. What is interesting is, chemically speaking, this system needs a photosensitizer that captures light and is the catalyst, and a cofactor that needs to have a precise redox potential. So here we have really a chemical information. And the interesting thing, we have discovered that this system only works if one specific nucleobase is there, oxoguanine, which is an oxidation, a stable oxidation product of guanine. If you take guanine, 2-guanine, 3-guanine, adenosine, cytosine, uracil, tyrosine, nothing happens. So we have a form of information. We have basically one functioning system over the 8 we have tried. Now, if this system was introduced in a system, this catalytic apparatus was introduced in a system composed of amphiphil, well, like this one, the question is, we will be able to make more amphiphilic molecules. So physically, what will happen is that we have a bead. I'll explain in a second why we have a bead here. And as you make more and more membrane, you force the excess of membrane to be released from the surface. And at some point, you got a small vesicle being formed. the second generation of the system. Why we use beads is two reasons. It's very difficult to recognize the original population if you don't have a beads from the second generation. They look alike. In this way, we have a physical way of separating the first generation, which is extremely heavy, from a second generation that is extremely light. So you use centrifugation. And if you look under the microscope, well, you start with beads that look like that. What we are looking is the fluorescence associated to the membrane. And over time, well, you can clearly see that membrane are spawning out of the beads, and you can separate them and end up having the second generation on one vials, the first generation that has been altered but contain the beads in another vials. And now you can ask the question, do both generations work? And indeed, the interesting thing is the first generation work, the second generation, either the altered bead system or the new free-floating generation are capable of performing the same type of function. So in a sense, there has been an inheritance between the two systems. And if you look at percentage and rate, you discover that actually it has been a very, very efficient function. Now, this is good. That's one thing. And one function, is it enough? We don't know. But we are in the process of reshuffling the same photochemistry to ligate DNA, and it works. So we could use basically a more complex system that has some nucleic acid that we could use as electron donor. You know, DNA are actually conducting polymers, and there are ways of making them give electrons. So that would be the next things. And if we take the more prebiotic system with Terence Key, we have figured out that not only we can use pyrophosphate to form amphiphyll, pyrophosphite, so that's P3 and not pyrophosphate, but we can at the same time make a small peptide. So here we'll go one step further, and I think the question is now one function we can somehow inherit, but is it enough to define a lineage? That we don't know. I want to finish and to conclude. The first part is a reintroduction, but basically chemical system can be basically formed to simulate complex biological processes of chemical for growth and replication. And in a very simple way, we seem to be able, from one generation of system to the other, to transmit a certain number of functions. So the hypothesis we will follow in the future is the system function and not its chemical composition per se could be considered as a form of inheritable traits whose transmission might have preceded the emergence of nucleic-based information apparatus. I want to thank my collaborator and obviously the guys in the lab that do the work, as well as the various entities that have financed this work, and thank you for your patience and attention.